Hello everyone, welcome to Biochemistry Rocks. Today we are going to be talking about amino acid degradation and the urea cycle. When the body's needs for proteins or amino acid are met, amino acids are degraded. The first step is the removal of the nitrogen, and this process occurs in the liver. In the process, the alpha amino group is transferred to alpha ketoglutarate to form glutamate. Glutamate is then oxidatively deaminated to form ammonium ion, and this process is catalyzed by aminotransferase. In this reaction shown below, we are transferring the amino component to this part of the molecule to form this, and then we regenerate the alpha ketoglutarate back again. Now let's talk about the oxidative deamination, which is catalyzed by glutamate dehydrogenase. Anytime you hear of dehydrogenase, you'll be talking about transfer of protons and therefore electrons. This requires electron carriers, NAD plus and then NADP plus, and the process leads to regeneration of the alpha ketoglutarate, and the enzyme, which is the glutamate dehydrogenase, is activated when we have low level of GDP and ADP, and inhibited when we have high energy levels, ATP and GTP. The process leads to the generation of citric acid and glycolytic intermediates. So in the reaction shown below, we are transferring protons to the electron carriers with a deamination of the amino group to form the ammonium ion, and then we are regenerating the alpha ketoglutarate. Now let's talk about classification of amino acid by the intermediate produced by the degradation of the carbon skeleton. That is, after we take out the amino component, the remaining carbon skeleton, how the body is able to make good use of that. So the various amino acids, like this group of amino acids, form pyruvate, and then this group also form acetyl-CoA, this group form acetyl-CoA, and so on. You can see all these groups are forming intermediates in the citric acid cycle. This structure is just an emphasis on what I just said about the citric acid intermediate, just the structure demonstration. So we have the pyruvate being the ultimate for this group of amino acids. Now let's talk about the urea cycle. So now, once the ammonium ion is removed from the amino acid, the body must find a way to get rid of it. And the process leads to the formation of urea. So urea production takes place in the matrix of the mitochondria. And let me make a highlight of that. And the steps involve the formation of carbamoyl phosphate, and this first step is catalyzed by carbamoyl phosphate synthesis, meaning that it requires ATP. So the reaction demonstrates using bicarbonate and energy to attach the phosphate group to the bicarbonate, and then with the addition of the ammonia component to form the carbamic acid here, with a generation of inorganic phosphate. That is the other energy source used. So it requires 2 moles of ATP, and with the final product being carbamoyl phosphate. All right. So once carbamoyl phosphate is formed, in the cytosol, it combines with ornithine to form citrulline, and then citrulline combined with aspartate, and this reaction is catalyzed by arginine synthesis. Again, requires the use of ATP, with the subsequent formation of arginine here, and then we form citric acid intermediate fumarate. This is just overall demonstration of what I just talked about. So the formation of arginine and formation of ornithine, that the process that occurs between the two leads to the generation of what? Urea, and that is excreted from the body. Thank you for your attention.